Okay, welcome to RISE Learning Machines seminars. RISE is Sweden's public research institute, and I am Olof Mogren. I'm part of the computer science department, and I work on foundational and applied AI projects with applications in ecology and climate change. This meeting will be recorded, and if anyone wants to be removed from this recording, please let us know. Also, please check out the collection of great talks that we have collected on our YouTube channel. Today, I have the pleasure to introduce Tobias Andermann, who is an assistant professor at Uppsala University. He works in biodiversity, and he's dedicated to providing data and computational tools for combating the global biodiversity crisis of today. He works in the intersection of molecular biology, spatial ecology, and machine learning. And the topic today is spatial biodiversity modeling with remote sensing and AI. And with this said, I will stop my screen sharing and hand over the word to you, Tobias.
Yeah, thank you for the introduction, Olaf. And once I get my screen sharing going here, I'll get started right away. All right. So, yeah, as you already heard, we're going to talk about biodiversity modeling today, and so we work a lot with neural network models for this task. But before I really dive into the modeling aspects, I want to spend a little time of laying the foundations of biodiversity research and the reasons why we're doing it. Just quick check. Do you see my screen? That's fine.
Yes, yep. Looks good.
Okay. So to give a little overview of what I'm going to talk about today, so we'll talk about the global biodiversity crisis. I'm sure you've heard of it, and I'm going to drop some numbers and get an idea for how big of an issue it really is. Then we'll move on to an example or first implementation of a biodiversity model with potential to be used by decision makers. And then I want to spend the end of the talk, or the last third, talking about the bottleneck of biodiversity data, which is really sort of the main bottleneck in this line of research, I would say, and also a potential way forward of how we can move past this bottleneck. But let's start at the very beginning. So the global biodiversity crisis in numbers. First of all, when we think of biodiversity, maybe you think of sort of the large mammal fauna in Africa, and many people go there for ecotourism to see these amazing animals. But what many people don't know or are not fully aware of is that we had almost this very similar fauna actually also here in Europe and on all continents really until not too long ago. So this is based on archaeological evidence from a site in Germany, in central Germany. And this site happens to be around 130,000 years old, and they find remnants of all of these species there, ranging from elephants, from forest elephants, to bisons, cave bears, cave lions, also hyenas, many species we don't even associate with Europe or Germany at this point anymore. But this was actually the fauna that our ancestors encountered when they moved out of Africa, and when European ancestors started settling, so early Homo sapiens that got to central Germany or central Europe around, current estimates are maybe around 60,000 years ago. Now, just a few 10,000 years later, we only have these species of megafauna left in Germany. So you see already something drastic has happened here, right? And also keep in mind, these mammal species, they actually existed for millions of years beforehand. So really something drastic happened just in the very recent history here. And it's likely humans, even early humans, had an effect on mammal extinctions, on other species extinctions. And here's just a little summary of a study from my PhD, actually, where we compiled all of the fossil evidence of species and sort of tried to estimate statistically what are the likely extinction dates of all these mammal species that we know of their existence. And then we constructed the extinction rate through time just to get an idea of how much of an increased extinction rate do we currently have compared to a baseline of a pre-human baseline, basically. And what we find is that the extinction rate throughout the last 120,000 years globally for mammals, it started here at around, at the baseline. And currently, so at the year 0, it is about 1,600-fold increased. So this is our estimate of how much faster is the pace of extinction happening right now compared to baseline extinction rates. This is only for mammals, and it's also only based on species that have gone extinct already. We have done a similar study last year that we published last year for birds globally. And here you just see mapped on a global map where bird extinctions in the last, I think the last 5,000 years where they have occurred. And you see kind of just roughly with this timeline here when they happened in these places. So not only a lot of mammal extinctions, also a lot of bird extinctions, and probably also a lot of extinctions in other groups that we have hardly any paleontological or other data for. So just based on these few groups where we have a good understanding of their fossil record and their fossil taxonomy, we get estimates of at least 1,600 times increased extinction rates. But we also have to keep in mind when we're looking at past extinction, so we're looking at the effect of of humans that were not putting as much pressure on the planet yet as we are currently. So we're talking about hunter-gatherers, farmers, and also like the very beginning of industrialization in the last few hundred years. But if you think of our human impact today, we have a much more intense use of natural resources and of the planet. So here we just see some of the major drivers of extinction or of the biodiversity crisis, which include large-scale mining and habitat degradation, but also very importantly pollution of water, air, soil, chemical pollution, plastic pollution. There's so much strain that we put on our planet just when it comes to pollution alone. And then we also have the general overpopulation of our species and the connected resource demands. And also on top of that, it seems to be that way that the more endangered a species becomes, the more valued it becomes on certain markets. So we also have a lot of illegal trade with endangered species and their products, like the ivory trade, for example. So when we look at modern numbers, so not just at past extinctions, but the current status of the world's biodiversity, there has been this study that made a very sort of, in a way, a bit of a shocking calculation or estimation. So here we're looking at biomass and the distribution of biomass on our planet. And the estimates are that so if you took all mammals that live on this planet together into one pile, 60% of the weight of that pile would be livestock. So cows, pigs, mammals that we keep for production of food and other products. 36% of the biomass of mammals are humans, so just our species alone. And only 4% of that pile would be actually made up of wild mammals. So you see how the wild part of this planet has been pushed on the very sort of brink of the total space and volume that it could actually occupy. That's similar for birds, actually. So the same study did this calculations. 70% of all bird biomass is made up by domesticated birds, and only 30% are wild. Another way of looking at the current state of biodiversity is to look at extinction threat levels. And maybe you've heard of the IUCN Red List. So this is a framework where species are manually or by a group of experts assessed, and they determine based on population trends and population size how endangered a given species is. And depending on certain sort of check lists, they decide if it's least concern, near threatened, vulnerable, endangered, or critically endangered. So these are like the 5 main classes in this framework. And many species are also data deficient. That means we don't have enough data to decide on their extinction threat status. And here you see for mammals how many species fall in each category. You see most of them are actually least concern, so that seems good. But then also if you add up the species between vulnerable, endangered, and critically endangered, all of these are considered of having a a high risk of extinction. And here you see sort of numeric estimates that are associated with these categories. The worst category, critically endangered, is actually evaluated as a 50% probability of extinction within the next 10 years. So these are all very critically endangered species, as the name of the category implies. But when you see these numbers, you understand how critical. Of course, these numbers are, I mean, these are categories, human-made categories. There's not like a clear categorical extinction risk, but we can assume that these sort of describe the average species risk in each category, with, of course, some variation, and also the chance of turning trends around. So not everything that's critically endangered has to stay critically endangered, but we also have seen already in the last decades many changes of conservation statuses for the worse, or also often for the better, actually. But it's important to keep in mind when we look at the current rate of extinction and the number of 1,600 that I mentioned before. This was only based on past extinction. If we factor in also the damage of the current standing diversity, so basically the damage we have caused other than extinctions, and if we factor in these threat levels, we can actually simulate for the next 70 years until the year 2100. If everything stays the way it has been, we would actually reach an extinction rate escalation of a factor of 30,000. I mean, this, of course, is a bit of a theoretical exercise, but it gives a bit of a feeling for the severity of how unnatural the current extinction rate is. So a factor of 30,000 means as many species go extinct every year as normally would within 30,000 years, which is a very long time period. So this is not anything that, or you may be aware of some of this, and actually also it's starting to really enter the public awareness that we have a biodiversity crisis, a global biodiversity crisis. And this has inspired many large-scale political conventions and decisions in the last years. So there's actually a lot of momentum right now to do something about the biodiversity crisis. And the most important one probably is the CBD, so the Convention of Biological Diversity by the UN, which has made a decision that's binding to all UN members, or to all states that ratified this convention, which is basically all of the UN members, that each of those countries by 2030 needs to protect 30% of their area for biodiversity conservation purposes. So that's a pretty tall order, especially given the timeline of 2030. So that's in 6 years. We need to make sure that 30% of all of the land area in each country is protected for, or at least managed for, biodiversity conservation. So this has also in the industry, there's a lot of momentum right now in terms of creating a biodiversity positive profile. So a lot of companies show great interest in having some framework of being able to evaluate their biological or biodiversity impact, and then also being able to justify or measure what biodiversity compensation they can do by purchasing land and managing it with a biodiversity positive management strategy. So it's similar to the carbon offsets that we have already seen implemented in the industry. Now, biodiversity offsets is the next big thing, I would say, in the current industry. And it's a huge developing market worldwide, and there is a lot of need for frameworks and methods to actually measure diversity impact and also the current state of diversity. So yeah, the big question is that nobody really has an answer to yet. How can we actually get these numbers? How can we know what biodiversity loss we are causing with a certain impact? For example, if a forestry company decides to cut down parts of a forest or individual trees, what is the loss for biodiversity? And then even more complicated is the question, how can we calculate what gain a certain offset is expected to give us? So if we purchase this area of land and manage it for the next 40 years in a certain biodiversity framework, what number can we put on it as an offset for the damage that we did? So these are the big open questions, but even more basic, we need an answer. First of all, what is actually the current value or status of biodiversity for a given place? even that question is not very well understood, or it's not so straightforward as one would think. So biodiversity remains to be a real complicated issue, but one that there's a lot of pressure on right now to find solutions for. So if we look at Sweden, and the rest of the talk, I'm going to focus on talking about the Swedish context. Right now, we have about 15% of the land area that's protected with the biodiversity positive management. And this needs to double within the next 6 years, as per UN decision. So we very soon will be in the political pressure to essentially double the area of protected, the size of protected areas across Sweden. And what is going to be the decision basis of this is not very clear and not very straightforward. So we hopefully it will be a decision basis that actually uses some biodiversity metric as its decision criterion, and not just whatever land is the cheapest to purchase and check off the 30% box. So this is the big question right now, and something that I think research and the academic sort of environment needs to address and answer. Also, within the EU, we actually have political decisions that have been made in terms of biodiversity. So the EU biodiversity strategy, also with the same deadline of 2030, and it's largely aligning with the UN decisions, but also has a bit more specifically actually addressing the European old growth forests as a specific target that we need to map and that we need to protect these forests. And when we look at Sweden's forest, so out of the European prime forest or old growth forest, Sweden actually holds the large majority of those forests, if we exclude the Russian Federation as belonging to Europe. And the majority of this is actually unmapped at this point. So we have pretty good data in Sweden in general on forest management and management history, but still where exactly all of the old growth and high conservation value forests are is still largely is not mapped. Of course, individual forest owners know often where there are interesting parts in their forest, but there's not a sort of overview of this data. And sort of alarmingly, this old growth forest is actually disappearing at an ever increasing rate. So there's an estimate that in the last 20 years or so, about 1/4 of Sweden's last unprotected old growth forest has been logged. So there's a bit of this kind of closing the bag panic in the forest industry. I would say that because these conventions are coming and being implemented, that right now is the time for many to decide to go out and harvest what can be harvested before it's going to be under protection. And in general, the demand of high quality wood is also increasing on. So there is a lot of pressure on Sweden's natural and old growth forests right now, and not really until they're mapped, we have no way of protecting them. So that's basically what we're going to talk about now is a model that can help us with mapping biodiversity and conservation value, specifically for Swedish forests. And just the bottom part here of the slide is relevant. So what we need when we want to model biodiversity or conservation value is, on the one hand, we need some type of biodiversity data to train our model. And then we also need some kind of contextual information or environmental information to train a model on biodiversity data and correlate it with available environmental data, and then being able to make predictions anywhere in Sweden using those same environmental data sources. So we need environmental data that has a spread across all of Sweden, and we also need biodiversity data that has a fairly good spread across all of Sweden. So when it comes to biodiversity data, we actually have, when it comes to Swedish forests, a really good data set of woodland key habitats. Maybe you've heard of this, nyckelbiotoper in Swedish. So these are polygons that have been mapped by the Swedish Forest Agency largely. And these are basically qualitative assessments of the forest. And so it's trained experts that go in and decide this part of the forest shows high naturalness and shows indicators of old growth. And then they map, taking the coordinates where this patch of high naturalness forest ends, and map these on in one central database. And here you see some of these examples. So across Sweden, you see them, they're everywhere. So if you look at all of these little polygons, they have basically spread across the whole country. But then on a more fine scale level, here we have 1 kilometer, this line. So you see these are often very small patches of forest, sometimes much less than a kilometer in diameter. And we have about 67,000 of those that Skogsstyrelsen compiled. And we have actually additional data from large forest companies that also have been mapping those in their holdings. And then, so to be able to automatically find these sites based on remote sensing data and other data products I'll show in a moment, we also need actually data on the opposite class. So we need to have some data on what do disturbed or non-high conservation value forests look like. And those are the blue polygons here. These are forest areas that have been registered for logging in the last 80 years. There's also a big open database managed by Skogsstyrelsen that shows where forest clearings have been done in the last decades. And so these will be of different ages. Some of them are just recent clear cuts and they look like this. But then others are also clear cuts that are maybe 40 or 60 years old and the trees have already regrown. For example, here in the background, this also probably has been clear cut at some point in the last decades, but trees have regrown, but it still is very different to a natural forest because it's basically a plantation or a managed production forest. So we can, yes.
Could this be sort of an arbitrary line to say 80 years is when it's a low conservation value? Could there be instances when it should be lower or when it should be higher?
Yeah, so that's a good point. I mean, it is a bit of an arbitrary line, but it's based on domain knowledge. So I've been in contact with the forest agency a lot, and so 80 years is a good cutoff because above that you can already have forests that have developed old growth characteristics again. But this also differs vastly between the north, where growth is much slower and takes a longer time to recover compared to the south, for example. So there is anyway not like really one cutoff, but this depends on the forest type, the tree species, and the latitude of the site that we're talking about. So it's a bit arbitrary, but still we at least have a range of different forest ages here in the opposite class in the blue polygons. So we just want the model to basically learn examples of what low conservation value forests look like, and we know at least that nowhere in Sweden that 80 years are not enough to develop high conservation value. So we can at least know all of these are below our quality threshold. Yeah.
Great, thank you. There's also a question from Peter in the chat. It also depends on the type of forestry that was practiced 80 years ago. But you did say clear cut, right?
Yeah, well, I'm pretty sure that all of these polygons are clear cuts. And plus, I think 80 years ago, to my knowledge, clear cutting has been the method for the last decades. I mean, now there are some different or some sites where different treatments are being tested or implemented, but I think the vast majority of these is definitely clear cuts. Yeah, but of course that matters a lot. Well, we don't really know yet the definite answer which forestry method is the best for biodiversity, and also how long does it take for forests to recover given a certain forestry method. So these are also open questions that I'm not even addressing here, but here, so now we are using these to train the model, and and then we also have environmental data that we correlate with these areas. So for environmental data, we use remote sensing data products. So we use the Sentinel-2 data that's openly available for research or for everyone, really. These are daily satellite images with several spectral channels. And also one thing you can actually model from these is you can calculate the normalized difference vegetation index, NDVI, that shows sort of highlights a bit more where we have plant growth, as in the spectral frequency that indicates chlorophyll production. So this is what we also know from Google Maps and so on, this satellite or airborne photography. And you can see already by just looking at the top of a forest like this, you can see areas of the forest that have been clear cut. You can see also when you really squint your eyes, different height profiles in the forest. You can see some parts that have roads in them. So there's a lot of information in these images. But then importantly, in Sweden, we also have another data product, the National Airborne Laser Scanning Data. And this is actually, if you haven't seen this before, I really recommend, if you're interested in data, you can download this from Landmaterial and you can open it in QGIS, which is a free program. And it's really amazing. So we have like super dense scans across all of Sweden using this airborne laser scanning technique. So this results basically in a really, really detailed 3D point cloud across all of Sweden. This is all free and open data. And you can see, if you visualize it, you can see every single tree top essentially. And you can also see things like power lines, clear cuttings. So this is obviously really important data when it comes to modeling the forest, because you can not only see, as in the satellite image, sort of rough differences between forest areas, but you can really calculate things like tree height, biomass, and also the variation of those on the landscape. And because also what's essential here is that the reflectance spectrum, sort of these laser beams basically hit the ground, they reflect back to the sensor, and the sensor can tell whether it hit solid ground or whether it hit vegetation. So you can really build one product of this is also a really detailed elevation model of the ground of Sweden. And then you can really like simulate or remove the vegetation from it or put it back on. And so you can really get very detailed estimates of biomass using this method. This is what one of the data products looks like resulting from these laser scanning data. So here it's rasterized into biomass, and you see here just a large area of a part of Sweden, southern Sweden, where you see this detailed raster of biomass. And here, if we zoom in a little more, this is now a few kilometers. You can see how what looks from the satellite image just like forest. When you look at these data, you see actually clearly some differences of different patches in tree height or in biomass, and you can see sort of more recent clear cuttings. So you can see much more detail on the landscape and on the forest composition. So now here in this model, we use our annotated polygons and all of these different environmental data from airborne laser scanning, from satellites, and also from other data sources like climate data. And we build a convolutional neural network that can take all this multi-channel image that comes from these data sources and actually segment. It's a segmentation task, really, where are high conservation value forests. And it learns that from our training data here. And so the training data, if we look at one single training instance, it looks like this, where we have our map with our polygons. And then we decided to take a 128 by 128 pixel image, which is kind of a standard image size in machine learning. And the pixel size here is 10 meters per pixel. So we have a 1.28 by 1.28 kilometer image that we crop out. So here we just happen to hit some high conservation and low conservation value forests in this image. And then we create an annotation mask or basically label. No, it's not really mask. We basically convert this polygon data into just a 2D array that is used for training the model. So this is our training labels, so to say. And in this image, we have the areas, the pixels that fall inside the high value polygons, they are coded as 1. The pixels that fall into the low value polygons are coded as 0. And everything else is coded as NA, as no data, because we don't know, even though there's forest there, we don't know if it's high or low conservation value forest, so we cannot feed the model any information on that. So the model really only looks at the areas that are annotated and ignores the rest. And then for the same place, we can extract these different environmental predictors via API request from here we use entirely data that's available on Skogs Data Lab. That's a public database managed by Skogsstyrelsen that anybody can get access to. You have to just request access, and then they will send you username and password, and you can download these data yourself. And yeah, it includes satellite image data. That's this year, also tree height and biomass, and then some temperature data, elevation data. I think this is the elevation profile here. Also data on the soil moisture, and soil depth. So there are different data layers that are available here that have been compiled from remote sensing data products. And so we have, in our training data set, we have 210,000 or 211,000 almost of these individual squares. So it's a huge data volume, so a big amount of training data that we have, because since we have these polygons distributed over entire Sweden. So each of these 211,000 instances contains some annotated information from these data here. And then we have a model. So this is, it's called an attention gate UNet. So it's a segmentation model, a convolutional neural network based segmentation model that has been originally developed for interpreting CT scan images and being able to annotate different organs in these images automatically, so that you don't have to do it manually. And in a way, we have a very similar problem here. So we have a multi-channel image instead of a CT scan image, and we want the network to learn where is our organ or our high conservation value forest in this image. And so it goes through a few layers of pooling. So it gets downsampled in size and image size, but the number of channels can increase or decrease. That depends on how many kernels you define in each convolution layer. And then it gets upsampled again back to the starting what it's called resolution. And then also it has these attention gates that basically means it projects one of the layers, from the downsampling part back to the upsampling part. So this basically, there's a matrix multiplication here with one of these channels, and it has been shown that this helps in keeping the contextual information in the image. So because if you downsample, you reduce it to the necessary features in a way. But of course, a lot of information gets lost in this process. And it has been shown that for segmentation tasks, specifically, these attention gates help to maintain or to improve the ability to segment the target patterns. And so then it goes through these steps, and then given the weights of this particular iteration, it calculates the loss of the output image and then optimizes the weights until the loss is minimized. And again, this is done obviously for all of the training data, or at least the 80% of the training data that were in the training set. So this here is just shown one instance basically, but picture this over hundreds of thousands. And once the model is trained, we can actually apply it anywhere in Sweden, because all of these layers that we used, all of these environmental features, they are available with complete coverage across the whole country. So we can now go and put a grid over Sweden and extract any 1.28 by 1.28 kilometer tile, get the image data via API request, run our trained model, and then we get a prediction of conservation value. We plug it back in to our map and do this repeatedly until we have a whole area covered. And so here you see, for example, the model predictions over, I forgot now exactly the size of this area, but some tens of kilometers. And you see how it very clearly identifies areas of the forest that it thinks are high conservation value forests, and also it predicts along a gradient. But the predictions are to be understood as probability of high conservation value forests. So if you have 0.7, it doesn't mean it has 70% conservation value. It means it has a 70% probability of it being of high value. So it's just a small sort of subtlety, but often misinterpreted what these values mean. Yeah, also, by the way, feel free to interrupt me if you have any questions, as you already did.
Yes, sorry, Tobias Andermann here.
Yeah.
I just wonder, I mean, if you have the training data, is either the nickel biotoper, which is high conservation value forest, and probably old forest, and then you have the young forest. You're not training on old forest without values. Am I right?
Yes, you're right. Basically, yeah, it would also be, those are not really mapped sort of strategically, so we didn't really have access to those data. But yeah, I guess the more fine-tuned training data one could produce, the better the model could get in really narrowing down.
So my follow-up question is actually, have you looked? I mean, there are areas where you have the nickel biotoper, and then you have at least the forests that are surrounding the nickel biotope is more or less equivalent if you just look on it from in a satellite imager, for example. You can't really see where the key biotope starts and where the key biotope ends. Have you looked at what happens? Are the high natural value forests that you identify, are they going out into the similar areas just adjacent to the key biotopes, or are they not doing it? Have you looked at it? So that's actually my question. If you have been studying the spread of high nature value forest into the old growth forest that has not been identified as high nature value forest, if you understand what I mean.
Yeah, I understand what you mean. And so I have not strategically, or I mean specifically done any summary statistics on it, but we'll see actually on the next slide. It may answer your question actually, because so here we have the predictions of our model, and highlighted here is only those places where it predicts a value higher than 0.5. So the rest is just masked out. So everything that's highlighted here in red is where our model thinks there is a good probability of high conservation value forests. And here you see just anecdotally the annotations. And here, these are nickel biotopa here in a test set. So these were not seen by the model when it was training. And it's actually almost the other way around, at least in this example, what you suspected. So at least here, often the nickel biotope boundaries are a little too generous, it doesn't, because they're drawn by hand on a computer, but they don't follow the natural line. So I think our model actually finds a more true shape given the forest conditions than the nickel biotope that's often drawn all the way to the road or but it identifies them pretty well overall in shape and also in size. But then of course it identifies the areas that are not annotated. So here somebody decided the nickel biotope ends here, but in fact our model says it actually continues. It could be that I don't know what made them decide to draw the line here. But
have any plans to make inventories just to make the check? I mean, it would be very interesting, I would say, just to see, okay, if you make if you look at what species you have on the outside and what species you have on the inside of the nickel biotope, when so you have it both in your high natural value forest area and then in nickel biotope, and then you see are there any differences or is it not?
Yeah, that would be interesting to actually visit these sites and do some inventory and see. There would maybe be a follow-up project, or large-scale project that maybe Skogstrelsen would be interested in doing at some point. To visit these places and see, is there any justification for it continuing, or is the border was the border set arbitrarily? So yeah, it would be interesting to measure biodiversity, for example, or other indicators, because the way old growth or the way these nickel biotopa are inventoried, it's not only using biodiversity, but also just using qualitative assessments of the forest, like the amount of that there's dead wood present and that there are old trees and things like that, and also heterogeneity of the landscape. Oh, somebody said something. Okay, guess it was a mistake. Yeah, but does that answer your question, or do you have?
Yeah, absolutely.
But yeah, it would be very interesting to investigate this further. This, by the way, is like super recent stuff, so we are actually just right now in the process of submitting this manuscript, so it hasn't been published yet, actually. But yeah, we get over the whole test set an accuracy of 92.5% of pixel accuracy of correctly predicted pixels. I guess I would never expect this model to be able to reach 100% if it's actually doing the right thing, because of these things that I just mentioned, like the annotations are not necessarily following natural borders. So we would not expect our model to exactly reproduce these weird unnatural polygons, but instead it maybe does an even better job, but at the cost of some drop in accuracy. But since it has to rely on these environmental predictors, it can only see natural borders and not the borders that were drawn by people here. And actually, the precision of the model is actually much higher than the recall. The recall is not so great. So that means the precision describes, as you probably know, but how many of those pixels that were predicted as our target class as high conservation value forests are in fact high conservation value forests. And here we can see at least that when our model predicts high conservation value forests, it is usually correct with 95% precision. But it misses some areas that should be high conservation value forests according to our training data. But that can also be because it doesn't fill out the whole polygon sometimes. So this will obviously cost it some recall percentages if it doesn't fill up to this border. So in a way, this model could be looked at as somewhat a conservative implementation. So when it infers high conservation value forests, it's usually correct, but it doesn't make these predictions very easily. So maybe many more forests could be labeled as high conservation value forests than what our model predicts. But I would say it's definitely useful because you can rely on the predictions of the target class. And so in this implementation, we actually trained 5 different models based on the biogeographic regions of Swedish forests, because the forests do look different in these bioregions. And so when the model looks at our remote sensing data, it will see very different patterns of what is considered a high conservation value forest if you're in the south of Sweden compared to the very north. So it makes sense here to separate these into different regions and train independent models on the data of those regions. So here we have now a model that where you can make predictions over all of Sweden, and it's useful for mapping high biodiversity value or high conservation value across Sweden, and at least give us a current value estimate for forests in particular. So we have the potential here for this to be useful in early planning by decision makers. And it allows to sort of calculate maybe a biodiversity value also for these implementations of offset calculation and so on, at least seeing what is the current value of these places that a company is purchasing. But I mean, there's a bit of a sort of a big but here actually. So we're not really, when we say conservation value of forests, it's not really the same as biodiversity, right? So we need to kind of think a bit about how do we define biodiversity. And here it's just defined as a binary variable, but that is, of course, biodiversity is much more complex than that. There's not just high and low biodiversity, but there's a whole gradient and insane complexity of biodiversity that we maybe should somehow integrate into these measures. So the next step is naturally to address the biodiversity data aspect of these types of models. So we need Sweden-wide or global, eventually, data sets of biodiversity inventories, and those inventories exist. So we have many inventories of different species groups that exist, especially here in Sweden. And we need to find some good data sets there that we can use to calibrate our models. Usually these inventories are specific to some species groups. And so we need something like this where we have different values across Sweden, where we actually have a numeric value that we can model with these models. So this brings me to the last part, and I'm trying to keep this a bit shorter than the other part. So the bottleneck of biodiversity data, because even though there's an insane amount of data points out there, for example, here we see in the background the map of species occurrence data on GBIF, which is kind of the global version of Artdatabanken. And you see sort of how many, I don't know how many, what the scale is here, but we have a lot of data points all over the world, and especially in Europe and North America, we have very, very high density of data. So this is species sightings that have been reported by scientists, but also by citizens. Let me just go through this a bit faster. So the point here is just to give some biodiversity background, but I need to move on a little bit because we are running out of time. But I mean, this is one point I want to focus on here, is when we think of biodiversity and the data that's available, we first have to sort of keep in mind that most species on this planet are actually not even described yet. So this may be surprising to you, but estimates are that 13 to 18% of all species on the planet are currently described, and the rest is basically dark diversity, things we don't even know about exist. And these are species that maybe are a bit less attention grabbing, so to say. So species you may overlook even if you step on them. But they may still be very important for biodiversity. So things like different groups of worms or unicellular organisms. I mean, so much of that biodiversity that we can't even really see or identify. But when it comes to mammals and plants, and also amphibians and reptiles, they generally have most of their species assessed. But already when we come to insects, which are actually one of the most species-rich group, they're only 10 to 20%, according to current estimates, that are at this point assessed. So the rest, most of the insects have not yet been scientifically described. And you may wonder, how can people come up with these numbers? This is based on yearly description rates of new species. And in some groups, you can sort of see a flattening of the curve where not many more new species are being discovered, because we have reached sort of we have described most of the species, and you need much more effort to find new ones. Whereas in other groups, you kind of see new species descriptions every year, like dozens or hundreds of them, and there's no flattening of it. And nowadays, you can also tell by DNA evidence how many more new sequences you find in an environmental sample, for example. For fungi, this number is to be taken with a grain of salt. That's a very rough estimate, but we at least know that most species are undescribed to the vast majority of them. And even worse, for protists and bacteria, there's hardly anything known. So we need to kind of keep that in mind when we think about biodiversity data availability. And so the spatial data that's available on GBIF that I mentioned is, of course, only of those species that we know of, so that exists. So, but of this fraction here, about 62% of them have at least one occurrence in the database. And only 6% of them have any extinction risk information about them. So we kind of have a hard time actually, just based on available data, mapping and deciding what species are where and how important are they actually for conservation and how threatened are they. And what's even worse is the taxonomic bias in the data that's available. So 75% of this whole big database of biodiversity data is aves, which is birds. So most of everything that we know about where it exists are birds. And the rest of the tree of life only makes up this tiny little fraction of this of the databases. So, and I just like to do this sort of exercise here, or this is a short little overview of life, just to appreciate how little of the fraction of life birds represent. I mean, how small of a subsection that actually is. So here we looking at the tree of life. So all the major groups that exist on this planet, and this tree is basically scaled by genome variation. So you can tell how far apart these groups are from each other as a measure of how different are their genomes. And down here we have the eukaryotes. So these are all of the species with that have a cell core. And of these eukaryotes, most of the things are weird groups you've never heard about. So these are protists, unicellular eukaryotes. And then only the small little group here makes up the animals. And among the animals, most things are also, again, strange things like sponges, different groups of worms, invertebrates. But then we have the vertebrates here as one of these small subgroups. And within the vertebrates, we have many different orders, and one of them is the reptiles and birds. So to just sort of think how little of the total diversity this group that we mostly focus on actually represents. And on a good day, we also have good data on amphibians and maybe mammals. But again, this doesn't make a big difference in the whole picture. So we mostly focus on these very few groups that we have good data for, also plants and some fungi. But we ignore 99.999% of all diversity. So any of these inventory data that is available lacks most of the species. And so I'm just brushing a little bit here. So there is actually, we are just looking at the very tip of this iceberg, but we can get a much deeper insight using environmental DNA. So this is a method that we are currently implementing in our lab, and also in general in Sweden. There's much more movement now in the environmental DNA field. So this is a method where you take DNA from an environment, so from soil, from air filters. You can also put up insect traps and just sequence sort of everything that you find in there. And so you basically take DNA extracts from these different environmental samples, and then you put it on a DNA sequencing machine. And so this technique is basically just possible through recent developments in DNA sequencing, because now you can, for an affordable price, actually get a lot of sequence data for a sample, and you need a lot of sequence data to really sequence through everything that's in the soil sample, for example. And then you can map these to reference database, which there is a big reference database of all of the species that we have defined so far. And usually they also deposit a DNA sequence along with those species. So when you now take a soil sample and you get your DNA from that, you can map those DNA sequences against the reference database. You can see which species of those that are already described in the database you actually found in your sample. And you can also find a lot of sequences, the vast majority of them, that don't have a match, but you have concrete evidence that they are there. So these are undescribed species most of the time, or sometimes variants of existing species that are just a bit different. So this is a real good method to capture this hidden diversity. So from here on, we're really like moving toward calibrating our methods on eDNA data. The only issue is with that, it's still very scattered between different studies and regions. So there isn't like really big national eDNA programs yet. And there's also some issues with noise and bias in these data. And so yeah, the spatial and sometimes taxonomic coverage is limited, because people, even though theoretically you can get everything with these data, what people often do is they're only interested in mushrooms, and then they use specific regions of the DNA and primers that are specific to only mushroom DNA. So then you basically throw away all of the stuff that's not mushrooms in your sample and only sequence the mushrooms. So this helps in getting a deeper insight into your target group, but removes most of the data. But unfortunately, many of the samples that exist have this limitation. And there's also still a lack of standardization. For example, what sequencing technology do you use? How deep do you sample the soil? All of these different variables. So in our group, we're actually testing and standardizing field and lab work protocols and match them against existing inventories of some groups that we have good data for. So we kind of try to find a good standardized method to do these samples, and to also know how to sequence them and how to process them in the lab. And there are many groups, many research groups that are working on this, and there's already a lot of knowledge, especially here in Sweden. Actually, we have many research groups specialized in eDNA data that have already been trailblazers in this field. We also have a project on insects. So this one was for fungi. This is for insects. And so the vision here, or the plan, is to use these types of data, so these really deep inventories, to train deep learning models, a bit similar to the one we saw earlier, but models that don't do necessarily segmentation tasks, but that give predictions of regression models, basically, that give predictions of diversity in any given cell. And here's one of those projects that we have ongoing at the moment from my PhD student, Adrian. And he works on a data set that's a little limited. It's only 200 sites across Sweden, but we have time series for those areas from eDNA data. And he's using a model that uses different environmental data layers as image input and has convolutional layers, but also combines them with just simple numeric features in then a fully connected part of the network. And then we can regress and get estimates of species richness at different times of the year. In this case, this is a data set that's only limited to insects, but theoretically, there are no limits here with eDNA data of what organism groups you want to model. So to sum up real quick, we've talked about the global biodiversity crisis in numbers and seen that there is it's a very alarming state of the planet. We also have seen a first implementation of a model that can give us conservation value predictions. And we now looked a little bit at the bottleneck of biodiversity data and the potential way forward, which is eDNA and getting deep inventories of sites. So we do have the technology. We do have the sequencing technology and also the extraction and lab knowledge to get DNA from all of these data, from these samples. So we have the technology to fill our gaping biodiversity knowledge in a matter of years. So maybe in time to meet these biodiversity goals. And it can be as simple as just having a soil sample of a site, and we will get so much more data than we have currently available. And right now, the bottleneck is still data collection. We also have the models infrastructures, the computational capabilities to process these data. We just need more data at this point. So that's all for me. And here I'm just closing with a slide of our group, the Biodiversity Data Lab based here in Uppsala University, and the current members of my group. Thank you.
Thank you, Tobias. And thank you for this. The closing remarks on this the new collection of the new data, the eDNA. I understand you haven't yet started training models on it, but have you sort of verified your current models with some of this new data?
No, not yet. So we are still in the very early phases. Basically, we just started here about a year and 3 months ago in Uppsala University. So we're a very new lab, and just now we have our first data sets coming in from our eDNA sampling. So it's too early to apply them for specific testing. So we're still in the calibration part, basically, when it comes to these data. But there is, yeah, I mean, maybe other groups have looked into this already, some more. So, but I haven't really specifically used it yet for validating the model projections. But that would be a very interesting thing to do. For example, the question that came up about the high conservation value forests, and if measuring biodiversity within the annotated polygon and within the neighboring area that's predicted by our model to also be high conservation. If we used eDNA, we could maybe have a simple decision basis to see is it different or not, and calibrate some threshold of deciding that. So there's a lot of potential for that.
Great, thank you. Very exciting work and very important work. So welcome back, everybody. Hopefully we'll have a seminar in 1 week, but we haven't yet decided on a speaker then. Otherwise, the next seminar will be May 16th. Welcome back, and thank you all for today.
Yeah, thank you all for listening.